Okay, we are very proud to receive Professor Onur Mutlu. He does not need an introduction, of course, but for the sake of decor, he's a professor in ETH Zurich, and he also has an appointment in Carnegie Mellon University at the same time. His research interests are in computer architecture, systems, security, and accelerators. He got his, well, this is in his official bio, so I can say it now. He had a double major in his undergraduate in Michigan on computer science and psychology. And then he went on to UT Austin to work with what Matteo calls Papayeo. And he got his master and PhD from UT Austin. Afterwards, he started the computer architecture lab in Microsoft. And let me read the rest. He has held various product and research positions at Intel, AMD, VMware, and Google. I know that he and his group and collaborators, the research that they have has been very leading to implementations in various microprocessors, and he has an impressive list of awards. I would like to go over it. Intel Outstanding Researcher Award, NVMW Persistent Impact Prize, IEEE High Performance Computer Architecture Test of Time Award, McCloskey Technical Achievement Award, ACM Morris Wilks Award, Inaugural Computer Society Young Computer Architect Award, Intel Early Career Faculty Award, Career Award, Carnegie Mellon University Lab Research Award, and of course he has quite a large number of best paper awards and topics in our field. He's an ACM and IEEE fellow, and also we have this ISTA Hall of Fame in our field. It's the benchmark. Long years his advisor reigned supreme there, and he was getting closer to his advisor. Now he's number one on that list, and his advisor is number two. So I think I wish you happy competition with your excellent graduate students in the next 40 years. They are coming hard on your heels on the Hall of Fame. So without further ado, he's going to talk about 80 minutes or so on accelerating genome analysis.
Okay, thank you very much, Osman, for the kind introduction, and thank you very much for inviting me. This was a talk that was scheduled in kind of last minute. I was here for Matteo's 70th birthday, talking about something else, memory-centric computing. Oh yeah, sorry, I forgot about that. Okay, thank you very much. If you haven't heard me from the back. Yeah, thanks for coming also to this talk. I don't remember when I was here last time. It's at least been 3 years. And I gave a talk here in this auditorium on, I think, in-memory computation. Today we decided to do something different, genome analysis, which is also quite important, I think. And clearly there are synergies with in-memory and in-storage computation. Can you hear an echo or something not so good? Is this better? Okay, this is better now because I was too close. Okay, let's get started. Osman said I have 80 minutes. I'll try to abide by it, but I think some of you heard this talk before. It's longer than 80 minutes, really. But feel free to ask questions somewhere in the middle. I have a lot to talk about, but we don't have to cover everything. I guess just out of curiosity, how many people here are working on computer architecture? Okay, quite a few. How many people here are working on genome analysis, genomics? Okay, that's smaller, but still substantial. How many people here are working on both? Okay, that's good. There's a good intersection. Okay, so we're going to talk about both. But I will give you some background also on genome analysis so that, let's say, it doesn't look very unfamiliar. And this is going to be especially my personal journey for some number of years. We've been working on this topic. Let me see if it's not working, so I'm going to... Okay, still not working. So I'll do this basically. Oh, that's also not working. That's not good. Okay. Okay, now it's working. Okay, it was not focused apparently. Okay, So basically, we're going to talk about system design for bioinformatics. I believe this is a critical problem going forward. If COVID-19 hasn't taught this us yet, I think there's a lot more you will see in the future. It has large scientific, medical, societal, and personal implications. And this talk is about a very, very specific portion of it. It's a key step in bioinformatics, which is genome sequence analysis. And even within that, we're going to focus on a particular step, which is called read map. So there's a much bigger world out there, basically, that we're not going to talk about. But this step is very, very important, I would say. And basically, many bottlenecks exist in accessing and manipulating huge amounts of genomic data during analysis. We're generating a lot of data. I'll show you some of these data generators. And we want to do increasingly sophisticated things on the data. But our mechanisms, system designs are falling short, essentially. And we will cover recent ideas that accelerate read mapping, especially from my personal perspective, because we've been doing a lot of this work in this area starting in September 2006. And we have a lot more resources right now dedicated to this topic. So hopefully it'll be interesting. So what we were dreaming about 15 years ago was to have an embedded device that can perform comprehensive genome analysis in real time. Let's say within a minute. You give this device to a doctor, and the doctor analyzes the genomic data of a patient plus whatever they know, and they make a decision as to what drug to administer, perhaps in a life-critical situation. If you could make that decision within a minute, that's great. You can save a critically ill patient very quickly, for example. Today, you cannot do that decision within several hours at least, depending on the analysis, and usually within days. I will actually show you some papers that were written by medical practitioners in the field that talk about this. We'll get to that in slide 40 or something. So I'll give you some initial overview first. So basically, there are many questions you can ask. For example, which of these DNAs does this DNA segment match with? What is the likely genetic disposition of this person to this particular drug? What disease or condition might this DNA or RNA piece be associated with? Clearly, we have been dealing with in COVID-19. And there are many other questions. Clearly, some of them can be life critical, safety critical also, right? You could, if you have this device, you could give it to a person also, and that person can privately do this analysis without informing anybody else in the world. So this device can have a lot of implications. Clearly, we didn't have a clue at that time because this device didn't exist, but I will show you devices that were introduced 7 years later that became much smaller, and we will talk about that. And these are some example devices. These exist right now, and there's some papers that I will mention that we've been writing about it, and others have been writing about it. And the good thing is these devices can be embedded today. The problem is these devices are not intelligent. They can basically generate data, but they cannot do any analysis on the data. So we will talk about that. You have to ship the data somewhere else, usually to a very powerful data center to actually do the analysis, especially sophisticated analysis. For example, you captured the genome and you want to compare it to 1 million other genomes in the world. Well, this device is not capable of doing that today. Maybe in the future. Okay. So just to give you a few overview readings, if you really want to get started on this topic, we've written a bunch of overview articles recently. This is one example. We were invited to write actually some of these. This is one of them from 2020. If you're interested, you can take a look at it. I'll cover some of the basics, but if you want to go into more depth, we have more information basically. This is another article at IEEE Micro. And this is a more recent one that we wrote actually. This was invited for this, I think 10th year anniversary of this journal. And we actually took the time to write a very long paper to talk about how do we actually analyze genomes today and a lot of acceleration mechanisms. So this is the most up-to-date one if you're interested in looking at it. So this is the agenda that I have today. It's a bit ambitious. We're going to talk about the problem first. I'll do a lot of motivation. And then we're going to talk about accelerating things at an algorithmic level. And then we're going to talk about hardware level acceleration. Usually it goes together with algorithmic level, actually. We're going to look at algorithms and hardware together. And if we have time, we're going to talk about the future. But future will be kind of blended in all of this also. And feel free to ask questions, I said. So probably all of you have seen a genome. A genome is made of 4 bases, A, C, T, G, I'm going to call them. I'm not going to go into detail. Clearly, we don't have time for this. And clearly, it affects a lot of things we do. It's not everything, clearly, but this nature part of us or living organisms affects what we look like, affects our characteristics, affects our actions, and it can affect our vulnerability to diseases or particular drugs, etc. And we're discovering this still right now. It's an ongoing field. There's a moving target in genomics. And these are real, clearly, all of you have this, hopefully. But this is a famous one. This is from Henrietta Lacks. Her genomes were used to do a lot of interesting analyses some decades ago without her permission. And this actually enabled us to learn a lot more about genomes. So there are actually other things that we're not going to talk about, like privacy. There's a huge privacy issue with genomics. If this information gets revealed to someone else, they can learn a lot more about you. They can use it against you in many different ways, for example. So there are a lot of privacy issues over here, which we're not going to talk about. But basically, a genome is, you can think of a genome as a huge string of characters. And all of you have 3.2 billion, around 3.3 billion base pairs, what is called pairs of bases. And this thing may matter a lot, for example. The question is, what is that thing and what does it correlate with? Right? This is called a single nucleotide. And that single nucleotide may differ. And that difference may make a huge difference in terms of your vulnerability to, for example, high blood pressure. I'm going to show you examples. And every living being has these. Humans are not that special, let's say. There are things that have much longer genomes, as you can see over here. And there are things that have shorter genomes. So what is the goal in DNA sequencing or RNA sequencing? You want to get these genomes and you want to basically find the complete sequence so that you can do some analysis on it. You can ask the question whether I have a C over there in that particular position. Right? We want to find the complete sequence of nucleotides in an organism's DNA. The key challenge is, unfortunately, today, there is no technology that exists that can give you this complete sequence without any errors. All machines today are choppers. Basically, they take DNA, they chop it into pieces, and they give you a puzzle, basically. They don't tell you how these pieces fit together. Most machines don't tell you how these pieces are related to each other also. You don't get any information. You get these pieces and you have to piece them back together to form a full genome. The question is, how do you do that? And this becomes a computational problem because of this reason. Well, partly because of this reason. Read mapping happens, now you have to piece things together. And that's the idea of read mapping. So basically, you have this large DNA molecule. It gets chopped into pieces by some very powerful machines, as we will see. These are called DNA fragments, and we can also think of them as reads. These reads can be short or long, as we will discuss. And the task is to take those reads and map them so that we can figure out what they are, where they're coming from. We're going to talk about that. But basically, we have these small randomized fragments. And in this paper that I mentioned earlier, we talk about how this works also. But I'm not going to talk about the sequencing machines as much today. So you get the reads from the sequencing machines, and you need to map the reads so that you can figure out where you differ, what you're vulnerable to. I liken this to untangling yarn bolt. It's actually, the problem is actually a little bit more complicated than this, because whenever you sample DNA, you don't necessarily get only a person's DNA. Okay, maybe you sample me, you get my DNA, but what if you sample some random surface and you get some DNA? You get a lot of other stuff over there, right? So you get a lot of different DNAs, potentially from different bacteria, viruses, humans, whatever. And you need to really untangle all of them. And this is called actually microbiome profiling. People have done studies, for example, in cities, they sample stuff on the subway and they try to figure out what's going on. Is there a disease that's coming up? It's very fascinating, I think. So you really need to deal with this in real life. Okay, these are the choppers. These are some quite powerful choppers, as you can see. I don't think this is the most powerful. They're all different and they all produce data, genomic data with different properties. We're going to talk about that. And these are some other high throughput sequencers. You can see that more recently things have been getting smaller, but also big machines have their benefits. Usually these nanopore sequencing, these smaller machines are high throughput, but they have high error rates. These big machines are even higher throughput, but they have low error rates. So if you want to do whenever you chop the genome, you also get errors, basically. The machines also introduce errors themselves, so you need to tolerate those errors. Okay, so clearly the Human Genome Project was quite popular. More recently, people claim that it ended, meaning that people have actually understood the whole genome. I don't believe that that's true at this point yet. But this has generated a lot of understanding of the human genome. It was not that expensive compared to the money we spend on wars today, but maybe we should spend more on this. But basically, because of these sequencers, what's happening today is we're getting a deluge of genomic data. People have been sequencing a lot of genomes and trying to understand them. Let me look at this figure. This is a famous figure that people have been drawing. What this shows is basically, this is the cost per raw megabase of DNA sequence. It's reduced over here a lot with the introduction of high throughput sequencing technologies, the big machines, Illumina, for example. And it's reduced again over here, for example, with the introduction of nanopore sequencing machines. And it keeps reducing. You can see that the trend of reduction is much faster than Moore's law. So the transistor cost is reducing much more slowly than these machines' output, let's say. Okay, and you can also look at the projected cost per human genome. So human genome cost at the beginning of the human genome project was expensive. Today, you can actually sequence your genomes with less than $1,000. Those machines actually that I showed you, nanopore sequencing machines, are less than about $1,000, let's say. Okay, so what has enabled this is really technology. It's good to understand the technology because technology can always change, and it's good to look forward to the technology as well. If you're a computer architect, for example, the technology may change underneath, right? It's changed from changed to CMOS, for example, today, but it could change to something else in the future. Here we are in a similar situation. This technology is light-based. This is Illumina-based sequencing, for example. It's called many, many things over here. But usually Illumina machines work by chopping the DNA into small segments and throwing those small segments into a glass flow cell surface. And based on the interactions you get on the glass flow cell surface, you determine whether you have an AC or T or G over here. Okay, that's the idea base. It's a statistical process in the end. You try to recognize, based on the chemical reaction, what picture you get. And that's why you may get errors. But people are perfect at this process. So the error rates are less than 1% today in terms of whether you correctly recognize an A as an A or a C as a C, G as a G, and T as a T. So these machines are quite good today, and they're very high throughput. If you're interested, you can learn more about this. I don't have time to go over it. I've already said a lot of this. So the benefit is you get high throughput, high speed, and low cost. Unfortunately, you get short reads. So remember, the human DNA is about 3.2 billion base pairs. What you get is pieces of 150 to 300 here. And now the computational task is to figure out where those 150 to 300 pieces, every single one, is coming from so that you can reconstruct the 3.3 billion base pairs. And that makes things a little bit harder, clear. I already said this, you get lower error rates, basically. And one of the reasons this is tough is reads lack information about their order and where they originated from. This technology doesn't give you, okay, this 150 base pairs is coming after this 150 base pairs. That information doesn't exist in this technology. There are some other technologies that do labeling, and that you have limited information about the differences, and they make it a little bit easier to analyze, but they're not as popular today. So as I said earlier, I liken this as you have this DNA. Genomics people like these names, like FASTA file. That's how they encode things. But it gets chopped into these incomplete files, and your task is to reconstruct that DNA, basically. And you may not have a complete construction if you don't have a good reference, for example. Okay, so this is one other machine, as you can see. So newer genome sequencing technologies are actually producing longer reads usually, but they have higher error rates because they're not as let's say optimized today. Or for example, this technology it's called a nanopore technology. You pass the DNA. It's a completely different technology. You pass the DNA through a nanoscale hole, and while the DNA is going through this nanoscale hole, you measure the current perturbations that happen. And the current perturbations that you get is different based on the bases that are passing through this hole. So fundamentally different, but it's more error prone. The error rates are on the order of 10 to 15%. Meaning what you get after sequencing is wrong 10 to 15%. And you still need to find that C, that single base pair that makes a difference. So the question is, how do you do that? So that amplifies the computational task. What people do is they actually sequence your DNA not once. They sequence it 30 times, 40 times, 50 times, 100 times, 200 times, so that they try to build a confidence in terms of what your DNA really looks like. So it's a complete statistical process, as you can see. So what I'm going to discuss is repeated many times, basically. So there are other things that are going to be involved that I'm going to discuss. Okay, so these are some technologies generally categorized, read length. So these are the short read technologies. Illumina, accuracy is very high. These are the long read technologies. Accuracy is relatively low, as you can see, but their lengths are 50,000, sometimes close to 1 million, actually. And there are some hi-fi reads that I mentioned that are long and accurate, but they're extremely expensive today. But again, keep in mind, in the future, these may become the common technology if we can figure out a way of reducing their cost. We don't know yet. And if you, I have a lot of references over here. If you really want to get into the field, these references, I believe, are good references that talk about different technologies. Okay, so basically, I hope I set up the task. We have the sequencing machine. We first need to map the reads so that we can figure out what we're dealing with. And then you do even more analyses, like variant calling, to figure out what we're different compared to some other disease marker, let's say. And then this leads to medical advancement or scientific discovery. And we're kind of bottlenecked. And there are many read mapping techniques, actually. I'm not going to talk about all of them. We covered this in this recent survey overview article. We basically covered 107 read mappers that were introduced between 1988 and 2020. And this is a long paper, as you can see, 111 pages. But there's a shorter version of it in genome biology that you can find. So people are still working on this, basically. There's a lot. Of course, let's talk about why do we care, right? I may be talking about, hopefully it's clear. How many people are convinced that we should care about this by now? Okay. How many people are not convinced? Okay, so maybe I can go through this relatively quickly. Okay, so basically, if I give you a bunch of sequences, tell me where they're the same, where they're different. This is very useful for many different purposes. Even identification of species, for example. And this is my favorite example. Humans are surprisingly very similar to each other, even though we may look very different in many ways. Humans and bananas are surprisingly very similar to each other also, as you can see. Yeah. So there's a lot of genetic material that's very similar. And I think one challenge is, how do we actually get rid of the similar material that doesn't matter? People are actually working on this also. There's a lot of similar parts of the DNA. But how do you get rid of, how do you not look at it, basically? And how do you not look at it while ensuring that you're not missing anything that's important? I think that's a key issue in genomic analysis today. So this is an example that I showed you. This is a single nucleotide polymorphism, SMT1, SMT2. These are correlated with blood pressure. And if you want to learn more about it, you can read papers, watch lectures, etc. And people are doing genome-wide association studies, basically. This is a lot of statistical analysis again. They look at people with heart disease and without heart disease and try to correlate these, what is called SNPs, single nucleotide polymorphisms, with heart disease occurrence. And again, many different things exist also based on whether patients respond to some treatment or do not respond to some treatment. You can find information about these on medical journals, actually. They talk about it. And people have discovered the causes, or at least the genetic markers of these some diseases, and you can find them. There's a lot of good papers that talk about these things. I'm not going to talk about that clearly. So this was what I mentioned earlier. For example, you can see that you can personalize the treatment for critically ill patients, which really require care very quickly. But today, some of those analyses cost 2 days or 5 days usually. If you want it to be done in 2 days, which may be too late to begin with, that's extremely costly. Basically, we want to reduce that to minutes, or less than a minute, actually, as I said, ideally. And if you're really interested in these topics, this is a very nice survey that was published in 2019. It's a bit old now, but it covers a lot of the basics. It talks about how the structural variation, so genomics is called the structural variation. You have variations in genome that affect your vulnerabilities to things, and this is a nice survey that talks about it. Okay, there are other analyses that you can do. This is what I mentioned earlier, like microbiome. So given a bunch of short sequences that you discover somehow, can you identify the organisms? And this brings us to microbiome profiling. And this is, I think, very important going forward for identification of pandemics, for example, in the future. And people have done studies like this in New York City, for example. They swabbed a lot of the locations in the metro, and they tried to figure out what kind of bacteria is lurking around, what kind of viruses are lurking around in the city. And a lot of them are known, as you can see. They couldn't figure out, basically, a good chunk. And you can see the papers over here. And people have tried to do this at the global scale with limited success. So there's a lot of data, basically, that people have actually generated, as you can see. And these are nice papers. This is from 2021. And people are predicting the data is going to increase. This is a Twitter post. But we're already at the exascale, basically. With 200K microbiomes, which is not a lot, I would argue, you're already at the exascale in terms of the data amount you have. And soon we're going to get to all of these scales. We're going to go off the scale, basically. These machines have been very powerful. These nanopore machines, this is actually from 2020, January. These nanopore machines were one of the first machines that were shipped to China when the pandemic was going on over there. And we learned a lot about COVID-19 because of these nanopore machines. And there are a lot of papers that are written on this topic also, if you're interested. But this is not new. I mean, people have used relatively portable sequencers that looked like this at that time to do the analysis on Ebola, for example, in Africa. And there's a lot of nice papers that talk about that as well. So, okay, I think you're already convinced, but there's a lot of use cases that we have. We can understand genetic variation, species, evolution. We can predict the presence and relative abundance of different microbes in a sample. We can do rapid surveillance of disease outbreaks and develop personalized medicine. So there are many, many applications of this. But basically, we're bottlenecked by this read mapping step. We may be bottlenecked by other steps later on, but initially, when we get the data, we're really bottlenecked by this read mapping step to begin with. And these are some example numbers. So the sequencers today are very powerful. Our analysis machines are not powerful. That's the key takeaway over here. And you can read papers that talk about how long it takes to analyze one human genome. Basically, sequencers can generate a lot more data than what we can process today reasonably. And the main reason is we have these very special purpose machines for data generation. They're extremely good at what they do, even though they're not perfect. And we have these poor machines that are not good. They're not designed for this purpose, basically. They're not specialized, essentially, I would say. And one goal we have is to specialize this machine so that it can match what this is producing. But that may not be enough because there's a lot of data that's generated. So ideally, you would like this machine to be as integrated as possible over here so that you don't move the data. Today, that those red arrows are going all over the place. People are sequencing, I don't know, some virus in Africa. They're sending all the data to data centers in Europe, America, and wherever. Basically, we're generating a lot of data and we're overwhelming our data communication systems because of this also. And that's also not helping our latency, clearly. So if this specialized machine is integrated over here, we can do much better, in my opinion. And I think we have a chance today going into the future. Okay, this is actually interesting, but I use this exact same picture in the memory-centric computing talk. The situation is the same in essentially wherever we generate data. Machine learning, for example, video is another example. We have a lot of data generators in video, and data analyzers are these poor machines. So we need accelerators again. Maybe we need to do the analysis closer to the machine itself. Okay, so basically in genome analysis, we need to reconstruct the entire genome for many sequenced reads. And we're back to this picture that I showed you earlier. So let's talk about read mapping. So read mapping is the method of aligning these reads that you generate against a known reference genome to detect matches and variations. So you have a known reference genome. It may not be perfect, but you're trying to align these reads so that you can actually figure out how these reads match together. This is one process. There's another process called de novo assembly, which is harder. This is the method of merging the reads in order to construct the original sequence. You may not have an original sequence here to begin with. I'm not going to talk about de novo assembly, but I think it's fascinating also. But there needs to be a lot of algorithms that need to be developed before we talk about architectures over there. I'm going to mainly focus on read mapping over here. So basically, you have these known reads. You want to map them to a reference genome so that you have a good idea of where these reads are really coming from. Okay. De novo assembly, you don't have the reference genome, basically. How do you reconstruct this? Looks like black magic, but it's not actually. You use some information. Okay. So the idea of read mapping is to map many short DNA fragments or reads to a known reference with some differences allowed. We need to have some differences because reference genome is not going to exactly match the reads that you have for two reasons. One is you have errors in the read, and the second is you have variations, genetic variations. My reference is different from your reference, for example. And there's no single human reference that is good for everyone today, clearly. Otherwise, we would be all the same. Okay, so you have DNA physically like this. You chop it into reads, and you map it into reference genome. So why is this hard? These are small reads. Okay, I think I already said. Okay, this is the harder part, basically. So if you want to do microbiome analysis, for example, metagenomic analysis, you have genetic material, the yarn balls that we mentioned over here. You have many reads in text format, and you're really trying to match this to a reference database, many references. So this actually makes the problem even harder, as you can see. But we're going to talk about one of them right now, not matching to a huge database. Okay, so this was the read mapping execution time before things were accelerated. Most of the time, read alignment covered most of the time, as you can see, more than 90%. We're going to reduce that, but let's talk about what this read alignment is. This is the edit distance computation. And just to give you a pictorial view of this, this is the reference genome over here that we assume is going to be true. These are the sequenced reads. what we're looking for is matches that look like this. Clearly, these sequence reads over here are matching this part, right, with some differences alone. The way you do it is you could do base-by-base comparison. You could compare the reads to each location in the reference and figure out whether they're equal or not. And that's clearly the fundamental way of building up. But we define a metric called edit distance. Edit distance is the minimum number of edits that are needed to make the read exactly match the reference segment. And edits can be multiple things. Edits can be insertions to the reference genome, deletions from the reference genome, or substitutions or a mismatch between the reference genome and the read. So there could be different types. And these different types can have meaning also in terms of, so you want to capture these different types because they have meaning in terms of whether you are vulnerable to a particular disease. A mismatch is not the same thing as an insertion, for example. So how do you do the edit distance computation, a computation of two character strings? In this case, we're looking at Netherlands and Switzerland. Basically, you can see that these match in the Erland part over here and the T over here. There's a mismatch between H and Z over here. There are two mismatches over here. There's a deletion in Netherlands where the I exists in Switzerland. And there's an insertion in Netherlands S where nothing exists in Switzerland. Right? So these are the different categories of things. And ideally, we would like to identify everything carefully because they have meanings. In fact, sequencing machines are biased towards making deletions, for example, sometimes because of the way they operate. If you know the characteristics of the sequencing machine, if you understand what's going on, you can correct for the errors before or during the read mapping process as well. So this is important, basically. That's to say. So basically, you need to do this for every single read. Remember, we have 300 base pairs and we have 3 billion base pairs. We're doing that for every single read. So a short map, a read may map to many locations, clearly. How can you find all mappings efficiently? You want to find all mappings because you want to figure out which ones are the real ones and which ones are important in the end. You need to tolerate small variances. I've already discussed this. Basically, each individual is different. How can we efficiently map each read with up to E errors present? This E is important because ideally, genomicists want, or biologists want this E to be higher so that they can figure out the larger variations between humans. If this E is, let's say, 1%, okay, you get something. If this E is 10%, you have possibility to discover new things, basically. But unfortunately, as this E becomes larger, the computation becomes much worse. Okay. And then you need to map each read very fast. I hope I've motivated you for this. How can we design a much higher performance read mapper? Because so today, at least traditionally, the way it's done is using dynamic programming algorithms. Edit distance computation is done using dynamic programming, which is essentially slow. I'm not going to go through this. How many people have learned about dynamic programming in computer science? Okay, that's a good chunk. I'd recommend you study it because we don't have time to cover it if you haven't learned it. But dynamic programming is the way of really doing this edit distance computation, as well as other things that in computer science. And you usually think of these as going through a matrix. And you need to compute through the matrix to figure out what is the smallest distance between which you can map these two strings to each other. And that's the idea of edit distance. So if you want to simulate this with Netherlands and Switzerland, you basically look at all of these entries over here, and then you fill this matrix by going through the entries. And this basically tells you your edit distance, right? It's 4 over here, because you can go through as close to diagonal as possible by getting to 4. But unfortunately, this is actually not easy. It takes time. And as your strings become larger, you can see that the time actually increases as well. Okay, I'll go through this quickly. This is for a longer version. Since we don't have a lot of time today, I'm not going to go through the basics. And if you're interested, actually, there are a lot of interesting papers that are written on this topic. This is a nice paper that talks about why you cannot do this in strongly subquadratic time. And we also cover a lot of algorithms in terms of their computational complexity also. A lot of them are in the end using edit distance, basically. There's no replacement for edit distance so far, let's say, or some way of computing edit distance so far, even though we're going to see a lot of tricks. So today what we do is actually, we can reduce that time for alignments, meaning sequence alignment, for 60% to more than 60%. And we're going to talk about different ways of accelerating. So you can, maybe I should not talk about this one, but let's go into the how do we accelerate things algorithmically. So that was kind of the background on what we're going to do. But we're going to talk about state-of-the-art read mapper design also. This is also a very broad field. There are a lot of different read mappers. I'm not going to cover all of them. They all have, again, different trade-offs. Some of them are more sensitive. They can find all mappings. Some of them are less sensitive. They find all mappings. Some of them require large memory. Some of them require less memory, etc. We don't have time to deal with all of that. I'm going to introduce you a way of doing read mapping that's, in general, applicable to a lot of read mappers. So what usually read mappers do is they pre-process the reference into some structure. It could be a hash table. And they use this hash table as an index to map the reads. So they form an index in the end, and they use index to map the reads. They don't go through the reference genome every single time. It's kind of like a database. You have the index of the database so that you can map things to it, right? Or do queries on it. So we designed one of those read mappers, specifically this one. But I think a lot of them are similar in nature. So you divide, in this particular one, we introduced, we have this notion of k-mers or 12-mers. These are basically strings of length k. So you can basically enumerate all possible strings of length k in the alphabet of the genome, ACTG. And you can basically have a hash table that says where each string appears in the reference genome. So you pre-process the reference genome and say a string of all A's appears in these locations, in those 4 locations. A string of all A's and the C at the end appears here. This string never appears anywhere, etc. So that's your hash table base. This is your index. And you can see that this index, even for the formation of this index, may be very memory intensive, right? Okay. So this is one example. You do this once for a reference, and then you use this index to query. So you get a read. This is a 36 base pair read that was chopped from your DNA, for example. And what you do is you actually chop this read into smaller k-mers, and then query the hash table using those k-mers. Basically, you have all A's, and this is where the locations are in the reference genome. Now you go into the reference genome and check whether the read actually matches the location in the reference genome. This k-mer told us it's at 12. So let's take a look at it. At 12, you read the reference genome. This is what you get. And you compare it to the, this is the read. At 12, you read the reference genome. This is the read. I guess this is the reference genome. This is the read. And they are equal in this case. But to be able to find out that they're equal, you need to do the edit distance computation. That's the verification or local alignments. It's costly, as you can see. So this is called a valid mapping. So now you go to 324. You do the same thing. You read the reference genome. It looks a bit different. You compare it to the read using edit distance computation. And it turns out they don't match. Make sense? So you do this for every single location, which is not good, clearly. That makes it slow. But you need to do it so that you cover the entire reference genome. Okay, that was our first step, and that was the mapper. This is a very comprehensive mapper, actually. But then we wanted to accelerate these things. So the problem is these are very slow because verification and alignment takes too long to execute. At the time, it was 95% because of edit distance computation, essentially. And our goal was to speed up the mapper. And this is where the interesting thing starts at this point. So how do you do this? I'm going to go through a bunch of works which have the same overarching key idea. And the key idea is don't do the computation. Filter fast before you align. Figure out how not to do the computation as much as possible without losing accuracy. That's the idea. Make sense? Okay, so basically you can divide the sequence reads into 2 categories. And one category is too dissimilar to reference, so don't do any computation on them. Some of them, they're similar enough to the reference that I should really check something careful. So the key is that filter. How do you make that decision? What is too dissimilar and what is really similar? Ideally, most of them actually are too dissimilar to the reference, so most of them should go here. You would like to identify those carefully. But you don't want to identify something that's similar to the reference as too dissimilar to the reference because you may be missing something really important if you actually do that. That's the idea. So we want to quickly find out these red ones and filter them out without costly computation and focus the processing power on these that are really important in the end. Processing power could be edit distance computation. And that's what this paper talks a lot about. So let's talk about some examples of this. When we first started this, after designing our mappers and looking at mappers, our goal was to do some algorithmic software level acceleration, because before you jump into hardware, it's good to optimize your algorithms, right? And our idea here was based on this observation. Most edit distance computations are actually unnecessary. 1 out of 1,000 potential locations pass the verification process. Basically, when you go and profile the read mapper, you figure out only one of those comparisons that you do out of 1,000 are actually comparisons that you should do. They're valid mappings. They lead to valid mappings. Everything else leads to invalid mappings. So assuming that these are equally costly to each other, you can speed up the mapper by 1000x or 999x, right? If you get rid of all of the useless computations. So how do you do that, of course? You can get rid of unnecessary verification calculations by detecting and rejecting early invalid mappings, filtering, and also reducing the number of potential mappings to examine. And in this work, we did both. So the first observation is this: adjacent k-mers in the read should also be adjacent to the reference genome. And you can figure this out by just observing the index, looking at the index. And we're going to look at that. And ReadMapper can quickly reject the mappings. Observation 2 is some k-mers are actually cheaper. The distribution of all k-mers in the reference genome is not equal. Some are actually very popular, some are not so popular. Figure out the not so popular ones and examine only those because they have fewer location lists. As long as you cover enough k-mers in your read, no problem, right? Because you need to map the read in the end and figure out the least popular k-mers and try to map the read over there. That's the idea. And there's theory around it that I'm not going to talk about because we don't have time. Let's take a look at the first one. We call the first one adjacency filtering. And I think it's best understood pictorially. For a valid mapping, adjacent k-mers in the read are also adjacent to the reference genome. So this is our read, this is our reference genome. And you know the location list because that's in the index, right? So if you look at this pictorially, you should say, okay, this is probably a valid mapping because the reads are close to each other in the reference genome. Whereas here, I have A's here, I have C's here, but I don't have anything else that has ACT close to each other in the reference genome. And if you can figure this out, you basically don't need to examine any of these. You load the reference genome and do the edit distance computation only on that one. That's the idea basically. Search for adjacent location to k-mers location list in the index structure. If more than E k-mers fail, there must be more than E errors, and that's an invalid mapping. Remember, you need to take into account errors. That's why we have E over here. If you were not taking into account errors, this would be an even easier problem, actually. Errors make it always more complicated, basically. And as E increases, this becomes more problematic, basically. Okay, so let me give you pictorially. So you have this read, you have these k-mers chopped into 3 k-mers. You basically look at, let's say, the first k-mer, AAA. It says it's in location 12. Then we ask the question. I expect that CCCC should be around 12 further, meaning at 24. Do I get 24 for CCCC? Oh, yes, I get 24. So maybe it's a valid mapping. But I expect TTTT at 36. Do I get 36 in the location list? Oh, yes, it's there. So let me check and verify. I think that this is a valid mapping, potentially. And then you do the edit distance computation here. Nice. Now we look at AAA, the next read. The next read starts at 324 in the reference genome. I expect that CCCC will be at 336. Is it there? No, there's nothing close by, and the distance is very far, so I reject this. So I'm not going to examine this read. That's the idea of it. So this way you can eliminate 99% of the mappings according to our results. Well, I think somewhere around that. I'll show you later. But basically, you can eliminate a lot of them this way. Cheap k-mer selections, again, a very simple idea. Reduce the number of potential mappings to examine. We said that k-mers have different costs. Let's take a look at it. We sort the k-mers based on their number of locations and select the k-mers with the fewest number of locations to verify. Let's take a look at it pictorially. This is a longer read right now to make the case. These are the location lists of the different k-mers. This k-mer has 4 locations. This k-mer, this k-mer has 2 locations. This k-mer has 1,000 locations. This k-mer has 2,000. So you can see the red ones are very popular. You want to avoid them as much as possible. So in this read, assuming the error is 2, you can actually examine only the blue ones because that covers the entire read. So the cheapest 3 k-mers are those. Expensive 3 k-mers, avoid them as much as possible. So this way, you in previous work, you can potentially examine 3,000 locations. If you're unlucky, 3,004 locations. We can reduce it to only 8 locations based. Does that make sense? You just look at the parts of the reference genome that has fewer locations for the k-mers. Okay. So if you do this, we actually, this is purely software based. We ran on a real system at the time, and we see significant speed ups. You can get to 19x. We didn't optimize it heavily. I think if you optimize it, you can get even better, but we'll leave it at that. And the reason is we're reducing the potential mapping by about 99% basically. We're not examining 99% of the mappings that a baseline mapper examines. We're not losing accuracy also, because we're ensuring that we're not eliminating anything that's potentially useful downstream. And this is a summary of it. And these are all open source. All of these work is open source. That's one nice thing about working in this field. People, in general, open source things, very different from computer architecture. I think I would encourage computer architects to do that also. But That's the nice thing about genomics. So you can download it. Okay, so any questions? Yes, please.
Yeah. This distribution you have shown about the possible locations of each. In this example, they are very, very unbalanced.
Yes.
Is this the reality?
Yeah. So that's a great question. I mean, this is of course a cooked up example. This is one particular example. So reality, they're quite unbalanced. Yes. Yeah, it could be easily 1000x difference, basically. Let me put it that way. Maybe these location numbers are exactly not the same, right? But the reality is, yeah, quite unbalanced.
And you have so many instances with like 1, 2, 4, 3 locations.
Yeah, these 2, 3 are just examples over here, but usually they're larger. Yeah, they're larger. It's not 2. Yeah. But there is imbalance, basically. Okay, that's a very good question. This was just a cooked up example to show the concept, not the exact numbers. Any other questions? Okay. Oh, yes, please.
Question is, what about minimizers?
Mhm.
As I understand, it's a usual way to decrease
Yeah.
the number of methods.
Yeah. I mean, those are actually, we cover all of those in the papers, but I'm not going to talk about them because, yeah, we're working on some minimizers. Certainly, optimizing the minimizers is another way of reducing things. We have some words. If you're interested, we can talk about it offline. Okay, so another question is, there's one, how do we accelerate these things? And we actually have been developing some metrics to actually accelerate things. I'm going to give you the key idea relatively quickly. And the idea over here is, if we're doing string comparisons, and if 2 strings differ by E edits, which is the E that we discussed earlier, then every base pair match can be aligned in at most 2 E shifts of one of the strings. This may be a little bit complicated, but the insight is shifting a string by 1 corrects for 1 error, and we're going to look at that. And the key idea over here in shifted Hamming distance is you take an end of 2 E Hamming distances of 2 strings to filter out invalid mappings. The good thing over here is it's completely bit parallel. You can use bit parallel operations that nicely map to SIMD instructions. And this way you can build a filter that's much faster than a very popular like Myers bit vector algorithm with only a 7% false positive rate. So here we're filtering again. Filtering doesn't have to be perfect. And this is the fastest CPU-based filtering algorithm at the time. So let me give you the key idea. So you have 2 strings, Istanbul. You can figure out put your favorite city over there if you want. Clearly these match. So Hamming distance is good for computing the matches and mismatches. But let's take a look at what happens if you have 1 deletion over here. You compare these 2 strings with Hamming distance. What you have is essentially 3 matches and 5 mismatches. That's not good. So if you want to cancel out the effect of this deletion that you have, you can actually shift the bottom string by 1 to the right. That's the idea. And do 2 comparisons. You compare the reference to the read, and you get 3 matches here. You shift the reads to the right by 1. Now you get 4 matches over here. Oh, sorry. I don't know what happens. So let's take a look at it again. And then what you do is you get the XORs of both of these strings. Here, the XOR will lead to, okay, I'll just compute these over here. And then you end the XOR bit vectors. What you have in the end is only 1 mismatch that you have, and the rest are matches. So you count the ones. If the number of ones is greater than some threshold, that means that, or less than some threshold, that means that this may be interesting to examine. That's the idea over here. Again, this is a filter. It's not perfect. We're not trying to compute edit distance or anything perfectly. We're trying to figure out whether 2 strings match or mismatch to a similar enough degree that we should focus more attention on that. If they're totally different from each other, then we don't focus attention based. That's the idea. So basically, this enables highly parallel matrix computation. From the matrix perspective, reference is here, query is here, or read is here. And this is their Hamming distance, right? The yellow one is their Hamming distance, basically. And what we do is we basically can have 2 deletion Hamming masks. If you look at the next diagonal, up, this is 1 deletion mask, and this is the second deletion mask. By shifting, you're really looking at these parts of the matrix. And by shifting to the bottom over here, or left over here, you're looking at the insertion masks over here. Essentially, you can cover the insertions. So if you're E, as long as you have, so you need to do it for E insertion mask and E deletion mask so that you can cover for error E, essentially. That's the idea. And these are completely parallel. There are no dependencies basically. You eliminate all the dependencies. So that's the key idea. So I'm not going into detail, but the paper has an example. There's some magic that you need to do to figure out how to amend for random zeros, because some random zeros also appear because of the way things work, which I will not talk about. But essentially, you end all the masks you get after XORing, and you accept if and only if the number of ones is less than a threshold. That's the idea. And this is your filter. And this filter turns out to be quite effective. Okay, I've already showed all of this. This just shows the formulation of it. Basically, what you do is we call this a neighborhood map. It's really what you have is really a neighborhood map. What you're really trying to get is to figure out the consecutive sequence of zeros. So you can see that there's a long string of zeros over here, 5 of them, and then you have 5 of them in another deletion mask. In the original diagonal over here. Basically, you can think of these as 4 different diagonals you're examining. 1, 2, 3. Or in this case, sorry, is it 4? Yeah, it's 4. Okay, it's 5, right? Okay, sorry, it's 5. Yeah, it's 5. 5 different diagonals you're examining, and you're trying to find the longest sequence of zeros in any diagonal, and you basically switch to another diagonal that has the longest sequence of zeros after that. You can think of it that way. Okay, we're going to see this picture over and over. That's why I spent some time over here. And this can be processed in parallel in many different technologies, and we've looked at many different things. I'll show you an example but this was a CPU. Even on CPU with SIMD instructions, it's actually very fast, 3x faster than the prior work. But now this makes it bottleneck the filtering algorithm basically. Sequencing generates many reads. Pre-alignment eliminates the need to verify or align reads to invalid mapping locations. And alignment is performed only on those reads that pass the filter. And it turns out this new bottleneck is really the pre-alignment and filtering. Alignment is less of a bottleneck now. Okay, and that's the paper that talked about. Any questions? Okay, now let me talk about hardware acceleration, because this is now once you get algorithms to be more friendly to hardware, which I believe this particular one is very friendly to hardware implementation, actually, all kinds of acceleration, you can try to accelerate them. Okay, I've already covered all of this. So we basically want to filter out most of the incorrect mappings, preserve all the correct mappings, and do this quickly. This quickly part is really important, and that's where the hardware acceleration comes in. I've already said this, so I'm going to go through this. This is meant for a longer discussion, but we don't have the time. I've already shown this also. So we actually designed an FPGA-based alignment filtering algorithm that uses shift in Hamming distance, which essentially does what we discussed, but you need to adapt the algorithm to the FPGA. And this is the paper that introduces it. You can also download the source code and use it. Other people have used it. They found similar results. And the algorithm is essentially similar, except it's adapted to the FPGA. In FPGA, you can do a lot of these bitwise operations very, very quickly, very, very fast, and in parallel. Let me give you the quick results quickly. Basically, this is what gatekeeper occupies, actually. Your FPGA is too big for this, because in the end, you're bottlenecked by your memory bandwidth, memory throughput. You don't have enough things to put into this gatekeeper logic. And we compare to SIMD instructions. Clearly, this is better than SIMD instructions. The overall takeaway is you get 10x speed up in read mapping overall, if you actually include all of these optimizations. And this is also pretty available. We encourage people to use it, report results. It's a bit old right now. It's 5 years old. And we're going to talk about other ideas that actually make things much better, as you will see. Okay. So the key conclusion, I think, is FPGA-based pre-alignment greatly speeds up read mapping. And this can be integrated with a sequencer also. I think this is where the real benefits will come from later on. You still need to, the problem with the FPGA is it's still bottlenecked by data movement into the FPGA. If it's sitting outside of your sequencer, okay, it speeds up things, but not good enough, let's say, as what you would like. And in the sequencer, you can help hide the complexity of the details of the FPGA. Also, you can enable real-time filtering while sequencing, and hopefully you can do on-device genome analysis. We're going to talk about that more. I don't believe this is the best algorithm, so we need to search for more and more algorithms, both in terms of pre-alignment as well as alignment going into the future, and the entire analysis. So we have a bunch of other accelerators I'm not going to talk about. So the question is, can we do better? Can we do faster, more accurate, and more scalable pre-alignment filtering? And I think here, where we really need to think about is how do we match the algorithms and the architecture? That picture that I showed you in the memory-centric computing lecture remains over here. I'm going to give you 2 more filters that were developed by Mohammed, who was my PhD student, who's my postdoc right now. I think the key ideas over here are actually quite nice. Let's start with the first one over here. So basically, correct alignment always includes long identical subsequences. We don't want to process even the long short reads basically together. We don't want to process the entire sequence at once. The key idea is to divide and conquer basically. You divide the short reads into smaller elements and use a sliding window approach to quickly and accurately find all long identical subsequences, consecutive zeros. Remember, identical subsequences are consecutive zeros in that diagonals. And we find out that this actually improves performance significantly. There are very large numbers. Let me give you an example over here. This is actually from an old paper, nice paper. They basically compare two different genomes over here. And you can see that correct alignment always follows these long identical subsequences over here. Close to the diagonal, but not always. So the idea over here is we build this neighborhood map that we discussed earlier with the insertion and deletion masks. And instead of processing it in its entirety, we take a sliding window approach. Sliding window of 4 base pairs in this particular case. And then we basically find all common subsequences shared between the two given sequences. So the longest string of zeros, as you can see over here. So you count them, you get that this is the longest, and then you say that you basically store the longest subsequence in the bit vector, which is 0, 0, 0, 0 over here. And then you do a sliding window, you move to the next one. You do the same thing. That's the longest subsequences over here, and store things over here. And you keep doing it, and you fill it out over here. In the end, you accept if and only if the number of ones is less than a threshold, meaning it's potentially a valid mapping if the number of ones is less than some threshold. That's the idea. And doing this again is very amenable to hardware acceleration, as you can see. And sliding window size is important. So if your window size is too small, then you get false accept rates that are very high. So you need to set your window size to be nice over here. There's a lot of analysis in the paper that I'm skipping, but the important part is here you can actually improve your FPGA utilization and make things even faster. Okay, and that's the paper, and you can again find all the source code online. Now let me talk about one of the latest ones in this direction, which we call Sneaky Snake, which uses computation even further. Again, we're going to use the same observation. We already said this, but we're going to map the problem to another problem that's very well known and people have developed solutions to, which is the single net routing problem in VLSI chip layout. So basically, single net routing problem routes an input to an output with some obstacles in between. And you can use only some number of obstacles. That's the idea. That's the number of errors, basically, we're going to use over here. So I'm going to give you a picture of this. But basically, this is the fastest algorithm that we have today. So this is our neighborhood map that I showed you. It's not shown as a diagonal over here, but it's linearized as arrays, as you can see. But you can see the different upper diagonals and different lower diagonals. It's the main diagonal. Assume that you form this like we have earlier. You can view this as just looking at the zeros, right? Zeros are white, ones are black. Now, this kind of looks like a layout problem, let's say. So what you're trying to do is you're trying to map from as many whites as possible while going through as few blacks as possible. That's the idea. You're going to minimize the obstacles. Okay, the snake's entrance here. The first obstacle happens after 4 over here, so it can move up to here, and that's your first obstacle. And then you reduce your edit distance count, if you will, or error count. And then you basically figure out where to go next. And the longest matches happen over here. And then you get to the next obstacle over here. And then you figure out where to go next. In this case, you have 3 choices that are all equal. You pick one. And then within 3 obstacles, you can reach the exit basically this way. So that's the idea over here. And the good thing over here is you don't need to build the neighborhood map initially. You can build this on the fly completely. So you reduce the computation cost over here. So I like this one from that perspective. It's more optimal, let's say. But we're using a well-known algorithm from the past. And it turns out it's actually very, very hardware efficient also. If you have enough memory bandwidth, this can actually improve your performance even more than what we show. And we show basically you get significant improvements on CPUs and GPUs and FPGAs, as you can see over here. So this works for any kind of hardware, if you will, because it's very simple, as you can see. Okay, and we compare it to read mappers that are as you can see. Okay, any questions? I can keep going over here, but I don't know how much time we have left as well. Yes, please. Mom.
Yeah, so some of these actually you can build this because you're using edit distance. You can 0 on 1 if it's a match or some kind of mismatch.
Yes.
There's some other, especially in the mappers, some use kappa fine system.
Yeah.
How do all these translate when you use kappa fine distances?
Yeah. So that's a good question. I mean, I think you can translate it somehow, but we didn't do that. And we didn't translate it. I believe the fundamental principles you can translate.
It would be like much more confident.
Yeah. Yeah, sure.
Is there any work on that? Or in practice, but it's always using it.
Yeah. So I think yeah, that's a good question. I don't know of any work that's done in that area. I think that's open to. I believe, yeah, there needs to be more work, frankly, looking at different metrics in the pre-filtering, even the pre-filtering step. Does that make sense?
Yeah, no, I'm just wondering if there is any why.
Yeah.
Maybe it's because the biologists think that it isn't good enough for.
So, yeah, I think a lot of people want to approximate the distance, that's for sure. But I think, yeah, I think part of the reason is there's not a lot of people working on this hardware acceleration, let's say, algorithmic and hardware acceleration. That's my feeling. Okay. Okay, so any other questions? Otherwise, I'll keep going. So basically, there's a lot more you can do. This is a more recent work where we wanted to actually build a framework to accelerate many different types of things. This can get closer to what you're thinking of potentially. But we didn't look at other distances over here. Basically, here we wanted to have a more flexible framework that can accelerate multiple steps of the genome sequence analysis pipeline. I'll give you the key idea over here. We start with a BiTAP algorithm. It's a fast and simple bitwise operations that can perform approximate string matching. It's actually an old algorithm from 1990s, 1992. And we modify and extend it with long read support. We wanted to have long reads over here. And it doesn't have traceback support so that you can compute edit distance. So we add some traceback support over here also. And we have some hardware accelerators. I will not bore you with the details of the hardware accelerators, but these are more general purpose, let's say, than what we have discussed so far. It's still specialized for the BiTAP, but I believe you can make it more general purpose. And you need different accelerators for bit vector operations as well as traceback. So traceback is fundamentally very different. So bit vectors are easier to do. Okay, I'll go through this quickly. So usually the hardware accelerator here looks like a systolic array so that we can maximize parallelism. It may remind you of machine learning accelerators. It is true, I think you need some systolic array to match the data that's coming out of the machines. Essentially, you need to match the memory throughput to the hardware accelerator throughput as well. And this is the traceback accelerator, which is a bit more complicated over here. In the end, it's simple logic, but it's more complicated than the other operations that we do. And these accelerators can be very low cost. This is a work that we did with Intel. A lot of the evaluations were with Intel state-of-the-art tools. You can read the paper for more detail. And you can see that the accelerator, overall hardware area overheads is much around 1% of a general purpose core. And there are many use cases where we can actually accelerate things. We've looked at multiple of them, and the paper covers them. For example, we can find the optimal alignments. We can do pre-alignments, like we discussed. We can do edit distance calculation, and we have other possible use cases as well that we didn't look at. But in the end, we get significant performance and energy improvements over state-of-the-art accelerators and CPUs and GPUs, of course. And I think there's more to do in this area. More recently, we've been looking at graph genomes. I think going forward, graph genomes are going to be even more important. And this poses even more problems. So what we look at so far is linear references, right? A reference is linear, that looks like this, but a graph-based reference encodes some of the variations in it. And people think that this is better because a linear reference represents only one person or probabilistically the population. But these graph-based references basically show what kind of variations may exist in a population. Humans don't look like a linear reference because if you look at all of the population of humans, you can basically trace your reference over here potentially. That's the idea over here. So here, when you do the mapping, you don't do the mapping to a linear match, but you do the mapping to all possible edges going out of a particular node graph, hop, let's say. And this makes things a lot more complicated. And there's no prior hardware design for this. And that's what we tried to do over here. And we wanted to have an accelerator that actually does sequence to graph mapping as well as sequence to sequence mapping for both short and long reads. Again, I don't have time to cover this in detail, but the hardware design you have is somewhat similar to CGRAM, but part of it needs to be different. You need to have a min-seed accelerator, for example. And we see significant speed up compared to existing alignment tools like Graph Aligner and VG and other things. So we can also do sequence to sequence alignment. And if you're interested, Damla has a talk on this that talks about the details. How much time do we have, Osman?
20 minutes.
Oh, okay, 20 minutes. Should we cover this or should we go into processing in memory? Or any other questions? Okay, so maybe just to give you an idea of the graph mapping pipeline, this is actually similar, and we accelerate a lot of the parts of it. But there, you need to construct a graph genome, and basically you need to do a lot of seeding, filtering, chaining, etc., to eliminate a lot of the things that you don't want to examine. And the difference again here is that if you have sequence to sequence alignment, you examine only the immediate, let's say, node in the graph because the graph is linear. But if you have a graph, now you have hops, and you need to do the computation across these hops also. So there are some other cells that you need to examine, and these number of cells grow with the number of hops that you have. Let me hide this over here. Okay. So we have an analysis of state-of-the-art tools if you're interested. This paper doesn't just talk about, okay, here's our accelerator, but we also analyze existing tools. And there are a lot of bottlenecks in these tools. You can see that a lot of the time, memory is the bottleneck, or either memory or the alignment is the bottleneck, as you can see. Okay. I think I'm going to skip a lot of these because we weren't talking about this, but you can look at the hardware accelerator in these slides. Essentially, this does accelerate minimizers, as somebody brought up over here. I think of minimizers as another way of filtering, right? Another filtering mechanism. So this is, you can think of this as a way of filtering the seeds that you need, so that you can figure out what to query in the graph and what to search in the graph. That's the idea. So you can think of this part of the accelerator as accelerating the seeding part to figure out what part of the reference genome you should really examine. And this is really how you examine the reference genome in the parts that you want that to be examined. So in the end, I think it all boils down to filtering, accelerating the filtering part so that you don't do stuff that is costly, and accelerating the costly part. So in the end, I think you need to accelerate both parts. That's my that's the way I view accelerating genome analysis in general. Don't do too much work. Accelerate figuring out how not to do too much work. And when you need to do a lot of work, which is the costly edit distance computation, for example, do it as efficiently as possible. That's the idea. Okay, and it needs to be done both in hardware and software, in my opinion. Okay, I'll not bore you with all these details since we don't have a lot of time, but this is the state of the art, and I'd encourage you to take a look at it if you're interested in it. And I believe graph genomes are going to be even more important. And again, we did this work together with Intel, and we find out that a lot of these analyses are based on their proprietary numbers. We can report only the numbers, not the models. But we find out that it's not that expensive in terms of area and power. And the results are, again, as I said, it's quite good. We get large factors or magnitudes of speed up. Usually, when you compare to software, the speed ups are much higher, as you can see. These are comparisons to software. Okay, I already said all of this. Okay, so let's now jump into what limits all of these actually. In the end, the CGRAM accelerator that we put, we put it under a high bandwidth memory because it actually benefits from this high bandwidth. And if you actually want to scale up the analysis to many, many genomes, we're still looking at a small number of genomes, one single genome mapped to one reference. It could be a graph-based reference. But if you want to scale up the analyses to hundreds of genomes, thousands of genomes, your memory bandwidth is going to be even worse of a bottleneck. That's why we've been looking at processing in memory and storage a lot. Essentially, a lot of these works are bottlenecked by DRAM bandwidth, especially the analysis that we've done. And processing in memory can actually alleviate this bottleneck. But the problem is now we need to design mapping and filtering algorithms to fit processing in memory. So the question is, how do we do it? And this is one example where we actually did this on a real system. We had this FPGA, which is coupled with high bandwidth memory. And we can put specialized logic into the FPGA to accelerate genomics. In this case, we put Sneaky Snake that we discussed earlier. And when you couple it with high bandwidth memory, you get a lot more performance improvement. So I've already said this over here. We want to perform read mapping near where data resides. And we carefully redesigned the accelerator logic of Sneaky Snake to exploit near memory computation capability of an FPGA that's uses HBM. And here we used an FPGA with IBM that has the OpenCAPI standard. I actually have a picture somewhere, but I don't know where it is. So we find out that actually the performance improvements are even higher. Performance energy efficiency improvements are even higher compared to an IBM Power 9 CPU. Yeah, this is the picture. In this work, actually, we not only looked at Sneaky Snake, but we also looked at some weather modeling application, and you see similar results. It turns out Sneaky Snake gets even higher performance than weather modeling. Okay, and you can look at the results in more detail. So this is real. Even on real systems, you get a lot more benefit today. And this is, I mean, it's processing near memory. Yes, you have an FPGA near memory coupled with very high bandwidth, and you get a lot of performance improvement. But imagine what you can do if you actually redesign the system such that you can do the processing inside the memory itself. We did this work, GrimFilter, a number of years ago. That was one of the first processing in memory-based genomics accelerators. I like it a lot, but I don't have time to go over it. Here, we really designed the filtering algorithm to fit processing in memory. I'm going to skip this one, though, unfortunately. But I'll send you all of these slides. You can take a look at them if you're interested. But I will talk about storage a little bit, because this is actually where things are going, in my opinion. Clearly, memory is interesting, but with memory, we can do only limited things. Why? Because, again, memory capacities are limited. They're going to increase, sure. But if you want to do something like a metagenomic analysis on, I don't know, thousands of genomes, they're probably not going to fit your memory. That's why we've been looking more into storage recently. And I think storage is quite interesting today. So let's take a look at, let's pull back a step a little bit. So we've been talking about a lot of stuff here, right? Main memory, caches, computation, accelerators. Okay, but data really moves from the storage or the device itself. I also think of the device that generates data. You can think of that as a storage, right? So there's computation overheads and there's data movement overheads. We reduced a lot of the data movement, a lot of the computation overhead using heuristics, accelerators, filters. That's what we've been talking about. We reduced the data movement overhead from main memory with in-memory processing. But the big elephant remains over there, which is a storage system. So you still need to move a lot of data from the storage to main memory to do the processing. So the key idea here is, can we do actually some work in the storage? It could be filtering. It could be the entire analysis. I don't know. Here we do filtering. We wanted to filter the reads that do not require alignment inside the storage system. Figure out what doesn't require alignment. Don't send it to the machine, basically, the main memory. And it turns out you can be quite effective at this. You send only a little over here. That's the idea. And I believe going forward, we should push this farther and farther into the device where the data is generated. You may not be able to do everything in the device. You still need to generate some data and store, but I think storage is very important. So here we actually designed two different filters. One is for exactly matching reads. If you can figure out the exactly matching reads quickly, which you can actually in the storage system, don't send them to the computation unit. Instead, send the results or somehow coordinate with the accelerator over here so that the accelerator knows that it doesn't need to process this. And the paper has the details on how to do that. Non-matching reads, these are reads that are not going to have a chance to match what we've discussed earlier, right? They do not have the potential matching locations, and you can skip alignment. Again, you coordinate with the accelerator over here so that the accelerator knows that it doesn't need to process. That's the idea. And again, we look at both short reads and long reads over here. We look at different data sets also. This is very much dependent on the data set. Clearly, what fraction of your data set is exactly matching, what fraction of your data set is non-matching. So the benefits that you get is very much dependent on your data sets. But there are reasons for this, basically. For example, exactly matching reads, you may have low sequencing error rates combined with low genetic variation. That's usually the case, actually, with high throughput machines that are fast. Non-matching reads, they do not have potential matching locations. You may have high sequencing error rates and high genetic variation, for example. Okay, I think I already said this. The difficulty in the design of this is workloads can exhibit different behavior, so your benefits may vary. And there are also limited hardware storage and resource in the storage system. So you cannot add a huge processor over there. Even though a huge processor already exists there, not a huge, but the service processor exists there, it's not as capable. What we did is we added accelerators near the flash chips at the flash translation layer so that they can exploit the bandwidth. And we have a work coming up actually at Micro that looks at doing this. Well, we don't look at genomic applications over there, but inside the flash arrays, you can do bitwise computations, just like you can do it in DRAM, which we didn't talk about. But inside the flash arrays, you can do even more bitwise computations on a larger number of blocks. And I think that can accelerate genomics fast also. Stay tuned for that one. I don't have slides for that one. But I believe that there's more that will happen, that should happen inside the storage base. Okay, I think I already discussed this. Basically, the paper shows that you get significant speed ups at low cost, and also significant energy reduction. And if you're interested, we have the paper at S+. There's also talks you can watch. Okay, so I believe there's more to do in memory. If you're interested in processing in memory, we can have a segue for maybe a next seminar in the future related to this. We've been doing a lot of work in this area. I'm not going to talk about processing in memory. But the good thing is, I think this is happening. So we've been doing work on the OpMem system. So basically, in my last seminar in 2019, I think I had the top pictures, but I didn't have any of these pictures over here. When I gave the seminar, I was talking about, okay, memory-centric computing is going to happen. Believe me. Now, I think the good thing is I have some real pictures to show you that it is happening. Hopefully, it will continue. Basically, in 2019, the OpMem company introduced the first general purpose processing in memory system. Here, you can have a general purpose processor inside the DRAM. I will show you examples of this. More recently, Samsung introduced an accelerator in DRAM again, mainly for machine learning applications. Potentially, genomics can make use of it, but it's very limited, I think. Here, genomics can make better use of it. It has its own limitations, but it's not as limited as this one, let's say. SK Hynix also introduced in GDDR6, for example, an accelerator in memory. Samsung and Facebook introduced the DRAM modules where they can do recommendation systems, inference faster. And Alibaba also introduced their chips, DRAM chips, to do, again, what they call recommendation systems. So I believe some of these chips can be used also for genomics, genome analysis. These are more specialized at this point. And also, they're not openly available. This is openly available. Well, openly meaning that you need to talk with the OpMem company and get access to it. And I believe you can, because they're interested in getting applications on their system clearly. And so basically, let's talk about this. So here you have DRAM modules. Inside each DRAM module that looks like this, you have processing enabled. These are processing enabled modules. What does that mean? That means that you have a DRAM bank. Next to each DRAM bank, you have a processor. And this processor is a processor that you've programmed probably, something similar basically. It's a multi-threaded processor. It's a general purpose processor. It has its own idiosyncrasies. It's only 400 megahertz, for example, because of the limitations of the DRAM substrate. It's not 4 gigahertz, but you reduce the data movement. You don't go off chip. So you can actually write your genome analysis or whatever program to accelerate your program, minimize the data movement, and map it across these different DRAM banks. So you can exploit very high level of memory parallels. And you can exploit locality also because there's an instruction cache and a data cache or scratch pad, as you can see over here. What you should not do in this processor is do a lot of expensive multiply operations, which you don't have in a lot of genomics applications. That's the good part. It's bad for machine learning because if you do 32-bit multiply, for example, or floating point, this floating point doesn't exist natively here in the hardware. There's an 8-bit multiplier. And also what you should not do is communicate across different processors, which is also potentially reducible in genome analysis applications compared to some other applications, for example, in my opinion. But basically, this may be a good fit for accelerating genome analysis. And we've been looking at it. We've been looking at some other workloads as well as genome analysis. If you're interested, Juan has been giving a lot of talks. He recently taught courses at Pumps Summer School over here. He's been doing that for years, actually. But he's been giving a lot of talks on this OpMem engine as well. But yeah, you can see that we've been looking at different applications over here, as well as genome analysis. And we release benchmark suites. If people are interested in working on that or contributing to benchmark suites, please feel free. We're very open. And there are papers also. Okay, I wanted to mention that. And we've been looking at machine learning on this, frankly, machine learning, some different types of machine learning, like k-means clustering, are actually very good over here. You can map it nicely. Neural networks, forget about it for now because of the multiplications. But some machine learning algorithms are actually very good fits. Genome analysis is also a very good fit. And we've been looking at some genome analysis also, and I believe there's more to do here. Okay, this is not necessarily related to machine, not necessarily related to UpMem, but we've been also looking at. So our goal, as I mentioned over here, is to integrate as much as possible into the sequencing device. And sequencing device, some of the sequencing devices, they do base calling, and we would like to do as much base calling and read mapping as possible inside the sequencing device. And this paper proposed an architecture that integrates base calling and read mapping and read quality control, which we didn't talk about as much in this talk, but inside the sequencing machine. And again, inside the memory of the sequencing machine. But I don't have time to talk about it. Okay, I think we can start concluding. So let me quickly talk about some future opportunities. I think I already talked about this actually. So there's one more new thing that I can talk about. So clearly new technologies, right? Nanopore was a new technology before we started. While we were going on, I think Nanopore became very, very interesting clearly, but there may be other new technologies out in the field. It's always good to keep an eye on those. Some of them may not be very good right now, but who knows? 5 years down the road, 10 years down the road. So I think this is very important in this field. But clearly making this technology better is also important. New applications, graph genomes, we discussed. I think that's very important. But there are also other new applications like reference genomes. People are questioning the reference genome now, which is good, I think. We've been questioning the linear reference genome. We've been generating new reference genomes. Reference genomes are getting updated, for example, with new information. The issue is if your reference genomes keep getting updated often, a graph genome, for example, how do you do those updates? How do you apply those updates? You've already mapped your data to a reference genome. When the reference genome gets updated, what happens to your mapping data? Do you remap everything again to the entire reference genome that gets updated? That's a lot of useless work potentially, right? Potentially useful work, but potentially useless work. How do you figure out what you should map and what you should remap? And I think this is a very important problem to minimize the overheads. And that's what we've been looking at with this airlift work, for example, which is under review right now. And also, while we were doing the airlift work, we developed some tools to map the constant regions between reference genomes much faster as well. This is already available. You can download and use it if you're interested. We'd be happy to support all of these tools if you shoot us email. Okay, I've already discussed newer genome sequence technology. These are actually some slides that talk about sequencing technologies, Nanopore in particular, but I'm not going to talk about that in the interest of time. Okay, COVID-19, I'm not going to talk about, but that's also interesting clearly. So let me conclude. Before I conclude, I should also say that things are happening in industry also. I will mention a couple of things. So Illumina has been developing their own FPGAs in their Dragon platform. According to them, they're going to integrate into sequencing machines. I don't know what the status of it is. They're probably integrating, probably not integrating. I don't know, but they're developing it basically. That's the good part. So hardware acceleration is getting into the machines. NVIDIA has been looking at these Parabricks, the GPU boards. And more recently, they've also introduced these DPX instructions, which are dynamic programming instructions, according to them, that accelerates dynamic programming. And one of the reasons was actually to accelerate genomics, but clearly dynamic programming is useful in other domains as well. So the good thing is industry is not, let's say, closed in this. I think they're going to look forward more. But again, I think this is an area where academia can lead because industry is usually not good at looking at the entire stack, algorithms to hardware, whereas academia, I think, is very good at looking maybe forward-looking and developing that entire stack. Okay, so let's basically pull back and recall where we are. So our goal was this: an embedded device that can perform comprehensive genome analysis in real time within a minute. Where are we today? Well, too far. Basically, we're not there yet. I think we have a lot of ways to go, frankly. Yeah, we have a huge memory on data movement bottleneck, energy efficiency, performance, they're not keeping up. And we haven't even talked about security and privacy yet. I think there's a lot over there. People are working on it, yes. But once you have stuff on a device, then this becomes a little bit different also. If you lose that device, what happens, right? For example, there's a lot of data that's related to you, perhaps. Okay, I think this is where we can conclude. I think we've already talked about this. I believe this is actually going to be even more important in the future. it has even more, it's going to have even more scientific, medical, societal, and personal implications, or people will realize it even more, let's say. And we've talked about just read mapping, which is one step in this analysis. There's a lot more here. And we talked about my personal journey, as I said, right? I didn't promise anything beyond that to you. I mean, I gave you some overview articles, which I would definitely recommend to take a look, but there's a lot more that's happening in the field that's very difficult to cover in our talk. I believe many future opportunities exist, as I said, especially with new sequencing technologies. You can still consider nanopore new, I believe, and especially with new applications and use cases. This is going to be even bigger than the technology. And you can see the examples over here. Let me acknowledge some people. Clearly, people who've written these papers are acknowledged. We actually organized a special session at ISBL site. Juan was leading it. That talks about acceleration in memory. If you're interested in in-memory acceleration, I recommend looking at it. And Mohammed and Gagan have been leading a lot of the bioinformatics work that we've been doing in the group. And we have a lot of detailed lectures if you're interested. We have a specialized genomics, accelerating genomics lecture if you are interested. We cover a lot of things, as you can see over here. A lot of things that I didn't cover over here as well. And clearly we have other lectures as well. And you can find them. We have a PIM course if people are interested in processing in memory. And we have heterogeneous systems and hardware software co-design course that are related to what I talked about. And as I said, storage is going to be even more important. So keep an eye on that. I'll acknowledge people who funded us. Thank you. And definitely my group more than anything else, because it's their work that I'm presenting. And these are some of the paces that you can assign to people who've done the work, let's say. And this is our latest newsletter. And as I said, everything is open source. If it's not on my webpage, because it may not be updated, it's on archive somewhere. You can find it. And if you cannot find it, let me know. And we open source essentially all of our tools. Most of our papers are associated with tools, and they're all open source on our GitHub. I think we made it to the end.
Right.
I'm happy to take any more questions.
Your talk was talking about computation. My question about the format of the format.
Yeah.
Because if I need to read 1 terabyte of file just to make short computation of the file, something very volatile. In my opinion, most is not all format out there.
Yeah, I absolutely agree. I think, yeah, I should probably in my next talk put something related to that, because yeah, I absolutely agree with you. I think we need to revisit everything we do in terms of formatting the data and preparing it for further analysis, because existing formats are very inefficient. And not only inefficient in terms of transfer, but also inefficient in terms of the analysis that you can do on the data.
I mean, like fast-minute indexing and
Yeah. Yeah, I think that's an area that there's a lot of opportunity. I absolutely agree with you. That's an area that we have not looked at. That's why I didn't cover it as much, but I will make sure that in the next talk, I'm going to have a slide about format. And also there's in the pipeline, there's a lot of format conversion that we have as well, right? Because people want to, let's say this is the problem of not optimizing the entire thing, but optimizing pieces. As a result, people convert from one format to another, another format to another, another format to another. And that conversion takes a lot of overhead. And if you really want to squeeze that to within a minute, then you're not going to be able to go through those conversions. I agree with you. We need to break the standard, basically. I think. Yeah.
Workflow. So we pass actually data from one program to another, convert data.
Yeah.
Huge part.
Yeah, sure.
Any other questions? I have a question about this. In general, these machines, you mentioned that you just chop into short reads. Did these guys look at how predictable or completely random that is, because it's on this falls on this glass surface, and if there is some kind of correlation, maybe you can do some sort of
correction.
Yeah, exactly.
Yeah.
Or correlation between the distances and where they fall into.
Yeah.
There's any kind of predictability.
Yeah, yeah. Sure. First of all, probably they have done it, but they don't publish. that's my answer to what they do. But I believe, yes, I think they analyze this a lot. That's how they got the error rates to very low levels. So I believe they've done a lot of analysis, and also a lot of the base callers today, especially for nanopore, they've been doing neural networks. So they do a lot of these statistical analyses in a way we don't quite understand, of course, to eliminate, to get rid of a lot of the predictive layers. I believe those neural networks are good at getting rid of these predictive layers for us, let's say, internally. Sure.
Yes. So, show us a lot of material. I think apart from technical material, we also have some talks about the devices for the PhD students and young researchers.
Yeah.
Basically, give us 2 minutes and what are the messages you give right to young people in the room. And the second one is a good one. So maybe you also can find your work in genome sequencing. Do the least that you can, and what you really have to do is doing precision, which is really the mindset of people involved. But it's also goes to Turkey.
Okay, let me answer the first one first. Okay, and then you'll remind me of the second one. Probably I'll forget by the time I answer the first one. I know. What advice do I have to PhD students, you mean?
Yeah.
Well, I think, well, I have talks on this one. You're right. I should put that talk. But I mean, I would suggest, basically, I think you're at a point in time where you can do a lot. I think there are a lot of resources today where people can learn. And as you can see, there are huge problems. I think we have huge problems in our machines today, computer architecture. We have huge problems in genome analysis applications today. My biggest suggestion is do your best work. Like, work hard. Work on what you're really excited about. And if you're excited about solving a problem, don't compromise. Go and solve the problem. Do as much as work possible and solve it. And while doing that, of course, I think you'll need to study hard. No question about that, right? Because there's a lot of work that has been done before you, but thinking differently, can put you in a spot that's very that can make a huge difference. Basically, I think the takeaway is you can make a big difference. But I think to be able to make that big difference, you need to have the mindset, in my opinion. And the mindset is, okay, I'm going to do something big here. I'm going to do whatever it takes to learn how to do that something big. And I'm going to do it. I don't know if this is the mindset of Balkans, but I think this is a good mindset for research, in my opinion. And you can do something big, basically. If you put your mind to it, we have a lot of problems where people can do something big. And that's what's happened. I mean, machine learning, for example, why is machine learning so exciting today? Because some graduate students and some students took classes on GPUs at the University of Toronto. And they said, okay, we're going to apply this GPU stuff that we learned for whatever they called ImageNet at that time, AlexNet, for example, right? All of those at that time. They wrote this paper in 2012 because they got inspired about this, right? These were just 2 graduate students, let's say. I said just 2 graduate students, recall. They changed the entire field. And their advisor. Don't forget their advisor, but usually the advisor is someone who guides and helps and enables you to make that big thing. Usually they don't have time to actually code the GPU, let's say. If they do, that's good, of course. Okay, I don't know what the other question was. Okay, I already answered that somehow. Okay. Okay, there was another question, I think. If you.
I have a question about it. I understand that then you create questions actually for reference genome. There are different techniques.
Yeah, there are many.
You take into account variants, no?
Yes, we take into account all of those.
You know, because what people think is just an internet, like shortest unique string, scientific probability, you actually avoid variants.
Yes.
There are a lot of these things there. There are some.
Yeah, I agree. Yeah, we cover a lot of those in our read mapping paper that covers 107 read mappers, let's say.
Yeah.
They categorize the different approaches, whether they use hashing, whether they use Bloom filters, whether they use something else, some other data structure.
You cannot tell your informatician if you didn't create your own.
Yeah, that's true. Yeah. And that's one of the things that we wanted to do in that paper, but it's not fully done. We wanted to basically analyze all of these different approaches and try to compare whether one is significantly better than the other or not. But of course, it's not very easy to do with 100+ mappers. We have some suggestions in that paper.
More obvious transformation based and
Yeah. Exactly.
and it's the same in compression. The always competition between
Exactly.
Okay, it's time for break again.
Thank you.